Hello children, last class we saw about the PCR method, amplification of gene of interest using PCR method and about the use of selectable markers. Today we will see about the foreign gene, obtaining the foreign gene product and how to produce the desired protein into large volumes. See, you know very well when you insert a piece of alien DNA into a cloning vector and transfer it in a bacterial cell or a plant cell or animal cell, the foreign DNA gets multiplied. And in almost all recombinant technology, the ultimate aim is to produce a desirable protein, and the foreign gene gets expressed under appropriate conditions. That is the desired gene. Once the foreign gene gets expressed under appropriate conditions, we have to produce the target protein on a large scale. Why it is necessary to produce them? It has got more commercial value. Okay, we are just doing desirable protein we are just producing it into large amount or quantities or cloning because they have got some value. Maybe it will be useful for an animal or a human being, or that desirable protein or a gene may be useful for a plant. So it has to be produced on a large scale. Okay, if any protein encoding gene is expressed in a heterologous host, see, heterologous host is nothing but basically the host will not be having the gene, that particular gene, but that gene can be introduced into the host. The desired gene responsible for a particular protein can be introduced into the host through a cloning vector. That is what heterologous host. Basically, it will not naturally, the gene will not be produced naturally inside the host body, but it can be made or it can be introduced into the host through a cloning vector. So that is the recombinant protein. Okay, and it can be grown in a small scale in the laboratory. This recombinant protein or the gene of interest, clone gene of interest, can be grown in the laboratory on a very small scale. The cultures may be used for extracting the desired protein and then purifying it by using different techniques. And the cells can also be multiplied in a continuous culture system. See, cells, you know very well, cells in the laboratory, it will be grown. This is a culture medium. The cells will be allowed to multiply in the culture medium. This is what the cell culture. See, this cell culture is used. The medium is in this the cell division takes place. And what happens once they keep on changing the medium, once the used medium is drained, and from the other side fresh medium will be added, because you cannot keep the old medium because the cell division cannot take place, it gets spoiled. So once one side it will be drained, other side fresh medium is added for the growth of the cells, so that to maintain the cell in the active or the log exponential phase, active cell division. That is what the log or the exponential phase, where active cell division takes place and the cells gets doubled. Okay, that is what this is what the cell culture. See, this is what the continuous they do in the laboratory. Animal cell culture, for example, is given taking a particular enzyme, culturing it, the primary cell. See, primary cell line, secondary cell line, and tertiary, all these subculturing they are doing it. Okay, and this is what, okay, animal cell culture in the laboratory. Okay, and the medium, fresh medium will be added and the old medium will be drained. That is for the growth of the cell, otherwise it will be spoiled. Okay, this type of culturing method produces large biomass, leading to higher yields of the desired protein. Okay, and small volume cultures cannot yield a high quantity of the product. So to produce in large quantities, we have to use bioreactors. Here in your book the picture is given of the bioreactors. To produce in large quantities, the use of bioreactors, where large volumes, in the laboratory only small volumes of the culture medium, whereas in the bioreactors large volumes, 100 to 1000 liters of culture can be processed. Okay, so these bioreactors are very helpful. It is a vessel like in which the raw materials are biologically converted into specific products. Okay, a bioreactor produce optimal condition for achieving the desired product, that is optimum growth condition such as pH, temperature, salts, vitamins, oxygen, everything. And here in your book it's given two types of bioreactors. First one is less simple stir tank bio and sparge stir tank bioreactors. Here you can see the curved base. Okay, first one is the curved base and this facilitate mixing and oxygen availability. Okay, so that it facilitate mixing and oxygen availability throughout the bioreactor. And the bioreactor apart from that, it will be having a agitator system to always to give fresh air and oxygen delivery system, foam control system, temperature control system, everything it will be having. This is another type, both for producing the desired product in large volumes, everything, optimum temperature, humidity, everything will be just given to produce it on a large scale. After completion of the biosynthetic stage, the product, okay, whatever the desired product, it has to be subjected to a series of processes which is known as the downstream processing. Okay, downstream processing, and these are the steps involved in downstream processing. See, this is what downstream processing, all the steps you can see. What are the steps? Anyway, I will just finally see it has to undergo purification and other things and all, separation, purification, everything, and finally it should be released to the market. Okay, so that is what downstream processing. The product has to undergo a series of processes before it is ready for marketing as a finished product. That is what we are going to see. Okay, downstream, what are the steps involved in downstream processing? There are only two steps, that is separation and purification. Separation, that involves filtration to separate the microbial cells from the liquid media. Filtration, which is followed by chemical purifications. Okay, products are impure and dilute, so it has to undergo purification, chemical purification. After that, the product has to be added suitable, it should be added suitable preservatives, and such formulation has to undergo clinical trials, as in the case of drugs. Drugs and all directly you just cannot introduce in the market. It has to undergo clinical trials, and strict quality control testing for each product is also required. It's very, very important. Quality control testing is very important, and the downstream processing and quality control testing, it vary from product to product. That is what drug for medicines is different, or any other product is useful for a plant growth, other things that is also different. So that's all about downstream processing, two things. So I think that is the end of the lesson, and the entire lesson what I have given, and these two diagrams are important, which you can practice at two different types. Okay, both are useful for producing the desired protein in large volumes, but only the shape is different, the names are different. And from the beginning till the end of the lesson, whatever notes I have given, please write in your classwork. And some of the questions you can just write from the exercises given at the back. Known questions you write. Okay, some of easy questions you can write. All these things you can write. Okay, this and all already it will be in the notes. All this like discuss, it is like distinguish or differentiate. This and all you write, and be thorough in this lesson, which is very important. Okay children, have a great day.